Unit 5, DNA. Look at the pretty picture. Oh, no more.
Not anymore.
Okay, a little bit of this is going to be review for some of our classes,
Which is good.
it's the basis, so it's very important. So inside of a eukaryote, it is located in the nucleus, that is DNA. The components that make up DNA are called nucleotides.
Those would be those monomers. If you go way back to when we were talking about organic chemistry.
And a nucleotide has 3 parts. The first part is going to be the 5-carbon sugar, which is right here. We call that deoxyribose. You have your phosphate group, that is here. And you have your nitrogen bases, which would be located here. You have 4 possible bases when it comes to the nitrogen bases. And this is what results in genetic variation in organisms.
You got adenine and guanine. Those are purines. Thymine and cytosine are pyrimidines. Anyone want to explain the difference between purines and pyrimidines?
Purines are double rings, so you got the 2 rings.
They can't see you.
Yeah, okay. I was going to grab it, but it's too far away. 1, 2 rings. So it's either an adenine or a guanine. You can't really tell. And then purines have a single ring, so they look like...
You said purines.
Oh, sorry, pyrimidines. Sorry, you guys can't see me pointing, but I point all the time. Pyrimidines have a single ring.
It's rude to point.
Oh, gosh.
Oh, my
That's
gosh.
beautiful.
It's a heavy ring.
Okay, you're not allowed to
It's
draw.
beautiful! But that's 1 ring.
Okay. Yay. Let's move on. Mrs. Hood already said the sugar and phosphate are what make up the backbone, so you will hear it referred to as the sugar-phosphate backbone. And then nitrogen bases are making up the middle. So if you think of it as like a ladder, okay, the outsides of the ladder, those are going to be your sugars and your phosphates. Okay, and then the rungs, or the place where you step on the ladders, those are your nitrogen bases.
Oh, hit it one more time.
One more time.
Or 2 more times, actually, because it shows you.
No dos.
Nitrogen bases.
Okay. Yay. Okay, it is double-stranded, which means there are 2 sides. Okay, it's 2 strands. I don't really know how to explain that one. Okay, and the strands twist to form a double helix.
That's as simple as that. No?
Okay. All right. Base pairing and sequencing.
A phosphate group and a deoxyribose sugar will remain the same in all nucleotides. So this right here, your phosphate group and your and your deoxyribose sugar are going to be the same in every single nucleotide. Now the thing that's going to vary is going to be your nitrogen bases, these things in the center or your rungs of your ladder. A always pairs with T, C always with G, and the same rule applies to all organisms. Then what is keeping those nitrogen bases together is going to be a hydrogen bond in the center of them.
Yep, and if you think back to when we were talking about chemistry, your hydrogen bonds are your weakest bonds, which is really important because that's what allows us to break DNA apart, is because they're the weak bonds.
And so you always want a strong backbone. So the sugar-phosphate backbone is actually covalently bonded together. So you can think of that as the strongest because you don't want DNA to just randomly break. But hydrogen bonds are weak, so it doesn't take much energy for them to unzip it.
Yep. Speaking of unzipping, we've got DNA replication. I'm just going to put everything up. Yes?
Replication is the process of copying DNA.
Yeah, you replicate it.
So one strand, so that would be a single strand, is going to serve as the template. Now a template is going to be kind of like your guideline, your blueprint of what the new strand will look like. Our rules for base pairing, which is our A to T and our G to C, are what are going to direct the replication and determine what the new strand will be comprised of. Someone else?
Okay. Each body cell gets a complete set of identical DNA. So it takes place before cell division. DNA serves only as a template. enzymes and other proteins do the actual work of replication. So enzymes will unzip the double helix, and this enzyme is called DNA helicase.
Okay, unzipping means we're going to be breaking those hydrogen bonds. Like a zipper, you unzip it, you get two parts.
Yep. And free-floating nucleotides from hydrogen bonds with the template strand.
They're going to be the ones that go in there. They add into there to make the new one.
Yeah.
That's what these
And I think the next slide explains that DNA polymerase is actually doing the job of bringing the nucleotides together. Yep. DNA polymerase is an enzyme that bonds the nucleotides together to form the double helix.
So helicase is breaking it apart, and polymerase is putting nucleotides on the single strands. And picture.
Right. Yeah, polymerase enzyme forms covalent bonds between the nucleotides and the new strand to form the sugar phosphate backbone. So in replication, your one strand that you begin with gets unzipped into two separate single strands, but then DNA polymerase comes in and places nucleotides along each part of it, creating two new double helix.
Now remember, your nucleotide is your phosphate and your deoxyribose, that's sugar, that's that backbone part, and then the nitrogen base, which could be either adenine, guanine, cytosine, thymine.
And it will match up to whatever one it's already given to determine which one it gets added.
So DNA polymerase is going to pull in a nucleotide that has thymine to match it with the nucleotide on the original DNA strand that has an adenine. So it's going to go through and it's going to be matching all of those nitrogen bases together because that's important. Okay.
Okay, two new molecules of DNA are formed, each with an original strand and a new strand. And DNA replication is semi-conservative. Semi-conservative means that it somewhat saves what it's already given, meaning that it has one new one and one old one, so it's conserved one old one and only made one new one instead of making a whole new set. Makes sense?
Excellent. Yep. Alright, replication is very fast and it's very accurate. Well, it's fast and accurate. I don't know if I can say very. DNA replication starts at many points in eukaryotic chromosomes, which means we don't have replication just starting here and then going all the way, this way.
Because that would make it very slow.
Yes, but we have multiple points where replication starts. And how does it start? Well, you've got that DNA, that helicase that comes in and starts unzipping. Okay, so it starts unzipping, and those points where it starts gets bigger, 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 until eventually you have two completely, well, I shouldn't say completely because it's semi-conservative, but you have two new strands. Each one carries one of the original templates, or one of the original strands from the DNA.
Yep, sounds good.
Oh, just kidding. DNA maybe someone else should say this.
DNA polymerase can find and correct errors. So basically, as it puts the nucleotides matching together, it also can proofread. So it's like if you're typing really fast, you can sometimes type the wrong letter. But if DNA polymerase was there, it would automatically tell you that doesn't fit correctly.
It's smart.
Yeah, DNA polymerase is very nice.
Okay, RNA is what carries the DNA instructions. The central dogma states, and basically that's just, I don't know how else to explain it.
Dogs from the center of America.
It's like a central, I don't want to say belief, because not belief, but accepted...
Almost, or...
An idea.
Accepted idea.
There we go.
By the majority of scientists.
It states that information flows from one direction, from DNA to RNA to proteins, meaning that it's always going to go in that order. Always will go DNA, RNA, proteins. It says that it also includes 3 points to the process, or 3 processes. Go ahead, because it adds them on the picture too.
Is it? Oh, all the way down?
It says that you're going to first have replication, then you'll have transcription, and then you'll have translation.
Okay, so replication is obviously where DNA is going to be replicated, what we've just talked about. Okay, transcription is when we're going to go from DNA to RNA, and then translation is when we're going to go from RNA to proteins.
And RNA is that link in between DNA and proteins. DNA cannot automatically go to proteins, so RNA is that central component that links them together.
Yep. Good?
Yep. Okay, and this is whenever we start to go in the structure of RNA. We talked about the structure of DNA, and they are similar, but there are some points that are different. First, with RNA, we still have that backbone that is a sugar and a phosphate, but except for in this example, our sugar is ribose. Our phosphate is still the same, but ribose is there instead of deoxyribose.
That's why it's RNA for ribose nucleic acid instead of DNA, which is deoxyribose nucleic acid.
Yeah. So RNA has 4 nitrogen bases, like DNA, adenine, cytosine, and guanine. But RNA, instead of having thymine, actually has uracil. So its sign is a U, is what you'll see it as. So in RNA, you'll see A's, C's, G's, and U's. I mean, I guess if you want to call that a U, sure.
It's a U. Oh, come on, Mr. Fuzzy Ribose. Deoxyribose, sorry.
RNA is a single strand. Instead of being a double strand like DNA, it is a single strand.
Yep.
Oh, we just went over all this.
Yeah, so these are just kind of a side-by-side comparison of RNA's difference from DNA. There's 3 major ways that you should remember. RNA has a ribose sugar, DNA has a deoxyribose sugar. RNA has uracil instead of thymine, DNA has thymine. RNA is a single-strand structure, and DNA is a double-stranded structure.
And just to remember always that this sugar is a 5-carbon sugar. Same thing with deoxyribose, but they're both 5-carbon sugars.
Mm-hmm.
Now what's going to differ is the oxygen
yeah.
where it's placed.
So it's like, yeah, that little oxygen's, I think it's just an
But
extra
you don't really need to
oxygen
worry
in
about
there.
that. You just need to know that it's ribose instead of deoxyribose.
Yep.
Okay, good?
Yep.
Yeah.
Excellent. Yay, now transcription. Transcription, should I put everything up there?
Yeah, go ahead.
That way they can see a picture.
Yep.
Transcription is going to be production of RNA from DNA. So we're going to have this transcription complex that's going to come in, and instead of adding in DNA to make a copy, it's making a copy in the form of RNA. Okay, it occurs in the nucleus, and it requires this RNA polymerase. Okay, instead of DNA polymerase, we're looking at RNA polymerase.
Because it's RNA.
What? RNA polymerase is going to bind to and then separate the DNA. And then it's going to bind RNA parts to make mRNA.
Which stands for messenger RNA. So RNA polymerase can do the job of
helicase.
DNA helicase and DNA polymerase. Just because it's a single strand, so it's a little simpler. You just need one of those enzymes. It can do the job for all of it.
And it starts the promoter, which is the part of the DNA with the specific sequence that RNA polymerase binds to.
It has a special code it binds to. Alright, transcription makes 3 types of RNA for protein synthesis. Mr. O'D already talked about mRNA. mRNA is messenger RNA because it carries the DNA message, or it carries that blueprint. Okay, that's going to be transformed or translated to form a protein. Okay, so it's the messenger. It's taking what DNA says and it's passing it on.
We're going to have to stop soon. Ribosomal RNA, which forms part of the ribosomes where the proteins are made. And then we have our transfer RNA that brings amino acids from the cytoplasm to the ribosomes.
That's about it.
Yep. Next step for the rest of it.